So where the science will come in today is understanding the real molecular mechanisms of life in normal growth and development, but also in circumstances where it goes wrong. Like in cancer, for instance, there are so many different molecular mechanisms that all lead to the same type of cancer. Take breast cancer as a phenotype, but that have different ways of becoming cancer. And it's only if we understand the molecular mechanisms that lead to that cancer that we will be able to find a target and treat it, to develop a diagnosis or a diagnostic, to find the real reason behind and to find a cure for it. Much more needs to be done to fully understand cancer. We are working within VIB. There are about 15 teams working on cancer, and they are all working on different mechanisms. For instance, we know that p53, when it's deficient, it will lead to cancer. So about half of the cancer have been detected with a mutation in p53 gene, but half of the cancers didn't have any mutation in p53. So for the time being, we are scratching the surface in terms of the drugs available on the market, but within a foreseeable time, there will be many mechanisms known, many targets identified for which drugs will be under development. Once you have understood the molecular mechanism of a disease, you basically have a clue or an avenue towards a diagnostic and a therapeutic. But in the future, the two will go together. The diagnostic to identify the molecular mechanism for this patient together with the drug that is treating that molecular mechanism for the disease. So once you know the molecular mechanism of a disease, you can start looking for a diagnostic. On the other side, we are convinced that many diseases have a kind of signature throughout the body where you can detect biomarkers that are part of that signature and which can provide us with early markers of something going on. I'm convinced that when you have a tumor, that other proteins, biochemicals are produced by these cells that normally are not produced by these cells. Well, in complex diseases where multiple genes lead to the phenotype, it might be more complex. And it might lead to getting diagnostic profiles rather than a specific mutation. But I'm convinced that basically the principles will be the same. It is understanding the mechanism of the disease and trying to manipulate it and to bring it back to where it should be, on the right path. So I think for complex diseases, the approach will be more difficult, more complex, but in principle will be the same. Well, the development of the technologies have been tremendously helpful over the last years. When I look back, for instance, to, let's say, 2008, 2007, 2008, it still took us like 20 million euros to do the sequence of a full genome of a human being. Today, 5 years later, you can get this for 1,000 euros. This is amazing development of technologies. Where it took us 5 years ago, it took us months to do it. Today, you can do it in 1 day. As I see it, within 20 years from now, cancer patients will survive on a rolling over therapy with different therapeutic molecules coming the one after the other and bringing them from one phase to the other, surviving in a kind of chronic mode, as you do today with HIV. The potential of sequencing with cancer is that one day we will be able to go to single-cell sequencing. Once we go to single-cell sequencing, where you can identify at single-cell level the mutations, I think that the signal-to-noise will increase and you will be able to indeed detect what is really a mutation and what is not a mutation. The genomics, the proteomics today is providing us is the different parts of the puzzle. But we don't know whether they are together, whether these parts form the complexes that you need to make them function in a cell. So what we need to see is not at the level of the genome, but at the level of the proteome. What proteins are there, whether they come together in the right complexes, whether they made the right complex structures that are doing the function, whether they are on the right place or not, and if not, what is wrong. And so I would say the development of bioimaging that is allowing us to see the complexes form and see whether they are functional or dysfunctional, this is a dream that we have as scientists. We are working on that. The technology is called CLEM, correlative light electron microscopy, and it will bring us to seeing how complexes of proteins are made and how they are functional or not. And then we will start understanding what is really wrong in the cell.